Hi. In the next two videos, we'll talk about DNA repair and what happens when DNA is damaged from external environmental conditions or when a mistake is made during replication. Whether a DNA change is a result of a mistaken replication or damage from the environment, they are both referred to as mutations. And as you know, mutations can have deleterious effects on health and cause various diseases. We'll take a look at the amount of DNA damage that occurs each day, which is pretty staggering. And then in the second video, we'll talk about 3 different ways of repairing 3 different types of DNA lesions. But first, we'll go with some background. So here you have a piece of double-stranded DNA, and a mistake has been made. The original DNA had a G, and there should have been a C over here, but instead an A has been incorporated into the strand. Now when it comes time for DNA synthesis, and these 2 strands separate, and both will be used as templates for the next generation of cells. When this A is replicated, it will be a template for a T base. This G will be a template for the proper insertion of a C nucleotide, which is what should have been there. And so that, in this case, 50% of all daughter cells will be mutated at this position, and 50% of the cells will be correct. This is in the absence of any kind of DNA repair. And we'll explore the consequences of this in a few slides. As mentioned, not only can there be mistakes made during synthesis, but there can be lots of damage done to a cell. This is an amazing table, which I saw in a talk by Joseph Shiloh, who's done a lot of work examining and quantitating the amount of damage done to a single mammalian cell in 24 hours. Over 70,000 mutations or damaging events occur every day to every one of your cells with their 3 billion base pairs. This is a staggering number in many ways. I had no idea this would be this large. The surveillance capability of your cell is outstanding. However, the cells are going to, on average, repair every single one of these mistakes. So our DNA repair machinery is phenomenal in its capabilities. I'd like to demonstrate the consequences of a mutation with the first molecularly defined disease, and that is sickle cell disease, or sickle cell anemia. In 1961, it was identified that sickle cell occurred because of a single nucleotide change of an A to a T. And this has the consequence of replacing a hydrophilic amino acid, glutamic acid, on the surface of a protein to a hydrophobic amino acid, valine. And we'll discuss in class how the single amino acid mutation causes the sickling effect that you see right here on these red blood cells. This is their normal shape, and this is their sickled shape. In principle, you already have enough information based on the lectures of protein structure to figure out the possible consequence of putting a hydrophobic residue on the surface of a protein. But let's discuss that in class. I also feel compelled to point out that it's been over 50 years since we identified the mutation responsible for the disease. Yet we do not have significantly improved treatments for the disease. Your prognosis is not that different today than if you get diagnosed with sickle cell disease as compared to when you would get diagnosed back then. There's greater support medical care, but not treatments that attack the underlying disease. And hopefully that will change soon, but the point I'm making is that you'll hear all the time in the popular literature, oh, a big breakthrough, we found the gene for this disease. But that can still be an incredibly long way to go before you actually make patients better once you know the disease gene. So you might ask, why have there been no improvements? And I would say that the short answer is that developing effective new treatments for disease is really, really hard. One of the ways to avoid having to correct a lot of mistakes or mutations is not to make mistakes in the first place. And the DNA polymerase does this by using several different mechanisms. Here on the top, we see a template strand and a growing strand. And as you can see, an error has been made. Somehow, a C was incorporated where there should have been a T base. This is already a very rare event, but it does happen. Now the polymerase is coming in and trying to add a second base. The polymerase is going to recognize that the base which it is trying to attach the new base to the 3' hydroxyl group is not correctly binding the existing template strand, and that most of the time this will be enough of a single to persuade the polymerase not to attach another base here. Instead, what it will mostly do is use additional enzymatic activity that it has a 3' to 5' exonuclease activity. That means starting at the 3' end and moving in the 5' direction, it can chew back bases that are not correctly base paired to the template strand. Once the incorrect base is removed, now it can add the proper bases and properly extend the DNA strand. Now this illustrates a very important point, which we will make clearer on the next slide. It's better thermodynamically to check 3 times pretty carefully than 1 time really carefully. For instance, you could think about how you want to spend your energy and time. You could be really careful about never putting the incorrect base there. Thermodynamically, that takes a lot of energy and a really long time. It's hard to do that when the bases themselves have a lot of molecular similarity. But instead what we'll do is we'll check 3 times. We won't very often put the wrong one there. That's number 1. Number 2, we won't add the next base if the previous one was incorrect. And number 3, we'll excise the incorrect base. So that's, in a sense, 3 attempts to get the proper base present. This slide quantifies how often errors are made during the various steps of the replication process and what the overall fidelity is. You can see up at the top that only 1 time in 10 to the 5th base pairs is an incorrect base included. Just to be clear, since sometimes people get confused, that's a 1 times 10 to the -5 error rate. Then, every time an incorrect base gets placed, the polymerase will catch it 99 times out of 100 using the 3' to 5' exonucleolytic proofreading. And for the 1 time that it sneaks through there and somehow a new base gets put on the strand, 99 times out of 100, that error will be recognized by mismatch repair machinery afterwards, which we'll see in the next video. So overall, in DNA replication, the mutation rate is 1 base out of 1 times 10 to the 9th bases, or 1 base out of 1 billion, is incorrectly included into the genome. Now your cells happen to have about 3 billion base pairs. That means every time a cell divides, you are introducing 3 new mutations to the cell on average. You might spend some time thinking about that. How come with all those mutations, we all don't have a large number of diseases? We'll discuss this a little in class, but it's interesting to think about. In class, we'll also look at some of the quantitative consequences of this trend for this error rate. Let me give you some numbers to think about. 3 times 10 to the 9th base pairs, 1 times 10 to the 12th cells, and for the sake of argument, let's consider a gene that has 1 times 10 to the 3, or 1,000 base pairs. Also, there are 3 times 10 to the 9th base pairs per genome and base pairs per gene of interest. The results can be pretty scary. In this slide, we see different locations where DNA can be damaged by the environment. You're not required to know any of these, but as you can see, the base pairs can be affected, the bond connecting the base pairs to the sugar can be affected, the sugar can be affected, or the phosphate group in the backbone can be affected. Damage can be done by oxidation, hydrolytic damage, or by the addition of other molecules, in particular methyl groups. One type of damage that we will talk about, which is one of the most common, is one caused by UV light, or excessive sun tanning, and it's called a Thymine Dimer. And it occurs mostly when there are 2 T's next to each other. Normally, T's have double bonds between these 2 carbons. But if 2 T's are next to each other and it absorbs an ultraviolet photon, you can form a 4-membered cyclic ring between the 2 bases. And now instead of having 2 independent bases, you have one larger molecule that's going to take 2 spots on the DNA. And your polymerase does not know how to process this when it's replicating the DNA. Fortunately, cells are pretty good at identifying this damage and repairing it. It's interesting to note that the thymine dimers are the exact molecular lesion which causes the body to tan. This was discovered at Boston University. If you take a cream of thymine dimers and wipe it onto untanned skin, the skin will tan just as if it were absorbing sunlight. And this makes wonderful sense. The sun is causing damage to DNA, and it's the damaged DNA that it is attempting to defend itself from the sun by producing melanin, a pigment which absorbs sunlight and offers protection for the DNA. Such creams are being developed clinically right now. You're responsible for knowing about this kind of damage. And now we'll break for the next video to talk about ways that cells will repair DNA damage or mutations.